Hi, welcome to Physionic. Welcome to the long form videos. Today we're going to be discussing something that's not really related to, as I've been discussing of late, talking about saturated fats and unsaturated fats and insulin signaling. Although I suppose in a indirect way we will be covering that. However, this is going to be to add some legitimacy to a concept that I think that a lot of people don't understand. That concept being, can we use animals or can we use animal cells as a proxy for what happens inside of our body, the human body, in human cells, as a proxy for human cells? And this came up because I've released a few videos now, quite a few videos actually, that have been in animal cells, specifically in a specific cell line known as the L6 cell line, which are rat cells, rat muscle cells specifically. And that certainly could draw the attention or this general understanding that why would we use animal cells if then we can't extrapolate that to us as humans? And I think that's a really fair question. I think a lot of people would get confused by that. And I think I'll say up front, I think that it would have been better for me to present the information that I did. So the studies that I presented on were the effects that saturated fat and unsaturated fat have on muscle cells. And these muscle cells, like I said, were specifically in animals. And I knew information that I did not present in those videos because I wanted to kind of let the story unfold over several different videos. And I think I think that was a mistake, because I think in the future what I'm going to do is to either go straight from like humans, like the studies in clinical trials, or still do this buildup of looking at the mechanisms, looking at how a particular treatment like saturated fat or unsaturated fat has an effect on cells, but specifically have those cells be human cells, primary cells or otherwise. And that's kind of what I wanted to address here, because although I knew this information that there is carryover, and that's kind of the big point here, and I'm going to show you some evidence of that there is carryover between, let's say, something as way out in left field as rat cells to human cells. So I'm going to show you some data. I'm going to go back to some of the data from the studies that I've covered. And I'm also going to show you some data from studies that have actually compared the two types of cells. And we're going to see where there are differences and where there are similarities. And kind of the big takeaway, really the question that I'm trying to answer is, can we use animal cells as a proxy for human cells? So add some nuance to that discussion. And for me to also just say that in the future I'll be focusing on human cells, but that doesn't mean that I'm necessarily going to disallow animal cells, because there are advantages to using animal cells that you don't get from humans, and I'll point them out as we go throughout. But if you're not familiar with who I am, my name is Nicholas Verhoeven, with that lengthy introduction, and I am a PhD candidate in molecular medicine, hold my master's in exercise physiology. I've been working in labs for I don't know, 6 to 8 years, something like that now, through undergrad, master's, and now through my PhD. So I'm definitely extremely familiar with working with cells. It's a big part of my research, and my thesis was in animals. Although I did do some human work as well that I didn't end up getting published on. It was for my exercise physiology degree. We had to do a series of different tests on people. But I digress. Let's go ahead and jump into this, and we'll answer these questions kind of as we go along. I'll be providing, like I said, some evidence. So the question that we're going to be examining, as I mentioned, is, can we use animal cells as a proxy for human cells? And this comes from information that I covered this paper. I actually covered another paper by this group as well, but I just wanted to throw up one of one of the titles. And you can see that it's in rat L6 skeletal muscles. And there were a lot of really striking results that were found on oxidative stress, insulin signaling, a series of different measures on the effect that palmitate, this saturated fat, had on not only mitochondrial health, but also on insulin signaling. And I reported the data, and I of course mentioned that these were cells, and I mentioned that they were rat cells, and that we couldn't jump to this conclusion of like, oh, well, obviously then, because we have this one study or two studies in cells, even if they were human cells, that we can then jump to this conclusion that saturated fat is not good for your health. We can't say that based off of this information, but it still starts to open the door of, one, the mechanisms, but also beyond that, gives us some idea of where we need to focus our attention when we are looking at human cells, when we are looking at what's known as in vivo models, so looking at actual living tissues and in situ, or looking at animal models, or looking at humans. So this is where things kind of started off, and I should have, like I said, I should have started with human cells. But that said, I had read multiple other studies at the time that I read this one, so I knew the results from this study did carry over quite well to some of the others. So we're going to look at a little bit of that here, but then I'm also going to be covering those studies in further depth later on. So then there was this paper that came out from, I forget what the journal was, but it came out, I think, maybe last year or the year before, and it actually literally compares the genome of and the gene scripts and the expressions of particular proteins like insulin signaling, metabolism, all kinds of different things between 4 different muscle types. So these L6 rat muscle cells, which were used up here, C2C12, which I have a lot of experience with because I use that for some of my master's work, and then human primary cells, and then human tissue, so the actual tissue itself. And it does a comparison between them. So if you're interested, you could certainly read that, and I'll show you some data from this specifically showing some differences and some similarities. Then this is actually in humans. This is in, or I should say human cells. And I wanted to cover this as well because there's going to be some interesting parallels. I'll leave it at that. And then this one tells us about insulin signaling between these very same cells, these rat L6 cells, as well as human cells. They do a comparison for the insulin signaling specifically. So all of this together is going to give us some good information that we're gonna base things off. Okay, so this first piece of data comes from this study right here that I mentioned, where they did profiling, and they're just looking at the gene sequences and looking at the amount of similarity between, hopefully you can still see that, it's mouse C2C12 right here, and then the mouse actual tissue right here. So C2C12s are muscle cells from mice, and they're what's known as immortal, but we don't need to get into that. The point is that you've got these different tissues or different cell types. Technically, they're all muscles, but they come from different sources: mouse, rat, humans. And then what they're just doing is profiling what's the difference between these genomes and the expression, not only just the genome, but the actual expression between the genomes. So let's say just as an example of this, let's say you have an enzyme like myosin, so you have a protein like myosin, which is commonly found in muscle. If you have a particular type of myosin, let's say myosin type 4, there's all kinds of different types of myosin. So myosin type 4, it might be found in all of the genomes of all these different tissues, but is it actually going to be expressed in all of them? So that's something. Also, it could just not be found in certain tissues. Like a mouse may not have myosin type 4, but they may have myosin type 6. Or humans might have myosin type 2. There are different types. So essentially what I'm saying is that it just compares between all of them and sees where's the similarity. So the more red that it is, the more similar it is. So the more white that it is, the more dissimilar that it is. And so here we've got human myotubes compared to human myotubes, which are the exact same thing. So of course they're going to have the greatest level of similarity. Human tissue has a lot of similarity to human myotubes. And that makes sense because, well, we're talking about cells compared to tissue. There's going to be a lot of similarity. It's not perfect, obviously. You see that it's not a complete one when you look at the value, the color key here. It's not a perfect one, but it's between, like, let's say 0.8 and 0.9, somewhere within there, or 0.75 and 0.85. You have a lot of similarity, but it's not exactly identical. Now, if you look at, for example, like the rat L6 cells to the human myotubes, which is some of the studies that I'm going to be pointing out, you see here that it's white. So you would think that, okay, well, then there's no similarity, but that's not how you actually read this. You read this as in, like, it has a great amount of similarity, but it's going to be the lowest amount of similarity between, by comparison to if you were to compare human myotubes to human myotubes. So yes, there's similarity. We'll say, let's say these are on a percent scale, so 70% similarity, but there's significant differences too. So figuring out what those differences are is going to be the ultimate goal, to find out if you can actually use these types of cells, these rat L6 cells or rat tissue or the mouse tissue or the C2C12 cells by comparison. So this kind of lays a groundwork of there are similarities and there are differences between these different tissues. And the more you go to a species, human to human, of course, you're going to have a lot more similarity than for rat to human or mouse to human or even mouse to rat. But even so, because the genome is so closely related to one another, let's say you have like 98% similarity, it ultimately comes out to, when you actually make measurements of different proteins and their interactions and things like that, it comes out to a pretty strong similarity. So like 70%, for example, or 80%, but it's not perfect. So then that raises the question then, okay, well, in what ways is it different? And in what ways is it similar? So I'm harping on this particular point because this is some of the measures that were taken in the rat study that I pointed out, this one right here. So I'm going to be focusing on insulin signaling. If we look at sugar uptake, so the uptake of glucose from the bloodstream, here we're looking at the comparison between these three cells. So we've got human skeletal muscle cells, which is right here. Then we've got our C2C12, and then we've got our L6. And you can see there's a difference, right? So question number one is, do they take up glucose? The answer is yes, they do. All three of them take glucose up at baseline. So this is without insulin stimulation. They just take up glucose, which, okay, so they have that similar in terms of what they do. It's not like rat cells just don't ever take up glucose, and using them for a glucose measure or a blood sugar measurement is makes no sense, because you're not going to find any difference. You will find a difference because they actually are involved in glucose uptake. However, the degree, what's known as the effect size, however, is much greater when you're talking about human skeletal muscle cells. And of course, it depends on which muscle cells you're talking about. But you can see, statistically speaking, that there is a difference between human skeletal muscle cells and L6 cells. There's greater similarity between the C2C12, so the mouse muscle cells, compared to the human cells when we're talking about baseline levels. So the big picture is here, one, that they all do the same thing, but two, the degree at which they do the same thing is different between the different conditions. Now, what we looked at in that paper, for example, was to look at insulin-stimulated glucose uptake, and here we find that there's a greater effect. So at baseline, there was a less of an effect when looking at the L6 cells. Now, when we add insulin, and do we find a greater delta? Do we find a greater difference? And there is a greater difference with the L6 cells compared to like the human cells or even the C2, C12 cells. So again, this means that those cells can be used as a measure of how does insulin affect the muscle cells. And the delta will be different, so the amount of change will be different, but they still do the same thing. So that's a big point to get across. Now, of course, with this, then another question might be, well, how do these fats that we investigated, how do they have an effect on this process? Is it similar to what we saw or what we might see in human cells? So this is something that I'll probably end up creating a standalone video for because all this other data came from the one study that, the whole study was just a comparison of the different muscle cells and muscle tissues. So this is, again, I realize this can be sometimes overwhelming. This is just a comparison, just a look between the two. So we're looking at protein levels of a series of different proteins. If you've been following Physionic for a while, you've heard of some of these proteins, like AKT, for example. GSK is glycogen synthase kinase, I want to say. Don't quote me on that. I'll have to double check. ERK is a signaling protein that's involved in a stress response. It's involved in other things as well, but we've been covering it as a stress response. You've got CREB. I've briefly mentioned it a few times in probably older videos where I've talked about metabolism or cholesterol synthesis. CREB is involved in that. And here what they're doing is they're using these cells. So these are all in rat cells. So these are the L6 cells all over here. All of these measures are in rat L6 cells, and these are all in human cells. Okay? And they've either not added insulin or they've added insulin through these different conditions, and then they've got these two different fats. So oleic acid and linoleic acid, I think, are the two, or linolenic acid, I can't remember. I think it's linoleic acid. And they've either added them or not added them throughout each condition. And then they just want to see, is there a change in these proteins? Is there a change in these signaling molecules? And what do we find? Well, if we just look at, let's say, the L6 cells, which is confirmed, this confirms some of the data that we looked at in the previous paper, which isn't a surprise. They're using the exact same cells. They found that when you add insulin, you get a greater phosphorylation of AKT at these activating AKT sites. And if you're not familiar with this kind of stuff, definitely check out my other content or my other videos where I actually explain this in far more depth. I'm just kind of essentially assuming that you know a little bit about insulin signaling at this point. But you have the total AKT levels right here. They don't budge much. Okay, so that's good. That's, again, consistent with all the other data we've looked at. And here we've got phosphorylation of AKT with the addition of insulin. This makes sense because you need to activate AKT to get proper insulin signaling. On the other hand, if I do remember this to be correct, like glycogen synthesis would be increased as well, so you get greater phosphorylation of the activation of these glycogen synthase proteins. CREB is another one. ERK is another one that gets activated. And here we're adding these different fats, and what do we find? Well, let's say when we add linoleic acid, for example, with insulin, then we see more robust activation of AKT, just as one measure, or more robust activation of ERK at these particular phosphorylation sites. Or we see greater synthesis, or we see greater CREB activation. And the same thing is seen with oleic acid. So the big point here is not necessarily to look at each one of these and try to get some sort of result out of it. The point is that you see changes with the addition of insulin, and then you get even greater changes with the addition of each one of these fats. Okay, so that's the point I'm trying to get across. That's all in these L6 cells. Now, if we focus our attention then to the human cells, where they do the exact same experiments, what do we find? We find the exact same results, that the the signaling is the exact same based on all of these measures that they did. So AKT, there's an increase in this phosphorylation of AKT when you add insulin. So no insulin versus insulin, it gets a little darker because there's greater activation. And when you add each one of these fats, you see an even greater activation, okay? This is all consistent with what we're seeing in these L6 muscle cells. The synthase, again, same thing. You get with greater insulin, you get more glycogen synthase kinase. And ERK, similar story. CREB, similar story. Now, here's where I'd like to mention that the advantages of something like using animal cells. So you might be thinking, well, why would I ever use animal cells if I'm going to get the same results in human cells? I'll tell you why. Because human cells are hard to get. When you take human cells from a human being, you take, for example, if you were to take primary cells, meaning that you're taking it straight from the human being, you would take a needle, you would, I don't want to get too graphic, but it's not that graphic, but I imagine some people sometimes get queasy, but you cut into, let's say, their leg, and then you use a needle and you snip a little piece of muscle. Now that piece of muscle may be something like 50 milligrams. It's a very small amount. And then you use that and you would add it to a plate. And then you would, what's known as trypsinize it. You would use a bunch of different compounds to break apart the collagen that holds the cells together in a particular uniform fashion. So you end up going through a series of different mechanisms to separate out the muscle cells from like the fibroblasts, from the collagen matrix itself. So you end up with not a whole lot of cells. And then you have to run, if we look back at this, you have to run experiments for AKT, for both phosphorylation, GSK, ERK, CREB. All of these require a certain number of cells that you need to run these experiments to be able to tell. And then, of course, with each one of these conditions as well. So you're using a lot of cells, and you can't just continuously be taking it from these individuals. So a really easy way to go to get around that is to get a proof of concept, for example, through animals. Animals, you can breed more animals without too much issue. And while I realize that for vegan individuals, this is a huge red flag, a huge problem. But with animals, you can just breed more, or you can just take more cells in general from them. And I don't want to get into all the ethics of that, but that's just the reality of how things go. So, and the reason why I'm pointing this out, not only just to make my overall point, but if you look at the quality, quote unquote, quality of these two data pieces, they're the same types of data. They're Western blots. They look at protein levels, which is the way that molecular biologists and cell biologists tell the amount of protein that's found in cells. You look at, for example, ERK. So here we get this really strong signal with these L6 cells. And here we see this really shoddy, just kind of, it looks kind of blurry. It looks dark. It just doesn't look very good. It's not to say that's not the fault of the researchers necessarily. It could be, but it's not necessarily the fault of the researchers. It's because they have so far fewer cells to use, so the background is much stronger. So they would need to take a lot more cells from an individual just to run a single experiment to get blots, or to get these bands that look just as crisp as these do over here, or these do over here. So there's a huge advantage in using animal cells in that you can easily just get more of them, and that gives you a much better signal. It gives you a better look at what's going on within the cells if you're just looking at things like mechanisms like we are right here. So, but this shows that insulin signaling is, in terms of the overall change, the delta may be different, as I mentioned here, right? The delta was different. So we saw a lower glucose level at baseline, and we saw greater glucose uptake with insulin stimulation. Well, here, maybe we'd have to quantify this more, but maybe you might get a greater effect in these L6 cells. But the general trend is that the human cells behave the exact same way as the L6 cells do. So this essentially what it shows is that it's a proof of concept that the L6 cells can be used as kind of a preliminary measure, never a situation where you take these L6 cells and then you jump to the conclusion of, oh, this definitely applies to humans, and saturated fat, or in this case, oleic acid, unsaturated fat, is highly beneficial to insulin signaling. It just gives you information, and then from there you would go to human cells, maybe if you wanted to do that, or you would go straight to tissues, or you would go straight to human beings and try to do these tests again. But it gives you an indication. And it's much easier to use animal cells because you can get more of them and it's easier to access and you can maintain them easier compared to human cells. So this is insulin signaling, all right, using the different fats. Now I'd like to cover just one more piece of data. All right, so on this final piece of data here, what I'm looking at is the exact same graphs that we looked at several weeks ago, looking at this particular study. Again, this one right here. And here they added the palmitate to the cells. Again, these are in the L6 cells. With and without insulin, so either basal or with the addition of insulin. And with or without the DNA repair enzyme, the human glycosylase that I mentioned. Again, if you're interested in more on this, just check out those videos because it's like an hour long. I don't want to go into all the details of everything again because I already covered it. So, and they're measuring the amount of 2-DG, which is 2-deoxyglucose, which is a way for them to actually measure the glucose uptake. They need to have a tagged glucose so that they can actually measure when the glucose goes from the media that the cells are held in. So the cells are in this dish, and there's media that feeds these cells. Well, if you just add glucose, well, then how are you going to measure it? How are you going to measure the glucose going from the media into the cells and where it goes within the cells as well? So you use a tagged glucose, something like 2-deoxyglucose, and that then allows you to track where this glucose goes. And here, all they're doing is just measuring the uptake. So the removal of glucose, the sugar, from the media into the cells. What we found is in the control condition that there was an increase with insulin stimulation in glucose uptake. And what was peculiar is that with palmitate addition, we saw a loss of that effect. So there was a blunting of the insulin effect when palmitate was exposed to the cells, palmitate being the saturated fat. So this was in L6 cells. So then, of course, then a question might be, well, is that true in human cells? And, well, an old paper that looked at this as well, and I have other papers that have also looked at this, but this paper came from human cells. And I just want to point this out, that even if we consider the delta differences and all that, it really seems that at least the overall pattern is the exact same. So the white bars are basal, so without insulin added. So the white bars are here, and you have an increase in the glucose uptake when you add insulin, which are the black bars. Then with palmitate, here we are again, we see that there is a blunting of that effect of insulin stimulation. And we see that there's also a blunting of the effect with another saturated fat. People have asked me if this applies to other saturated fats. I will be covering. I'm hoping to cover content on this. I will probably be covering this paper, which they look at stearate. And then with the addition of oleate, we see this research group released another paper where they looked at oleate as well, and they found these results. They found that oleate increased or rebounded the insulin signaling to the point, not only with this situation right here, with the actual signaling within the cells, but also with the rebound of the glucose uptake. And that's what this research group also found. I don't think they actually ended up testing glucose, but they did measure all the insulin signaling. And other studies have checked out glucose, and it follows the exact same pattern. And then the combination of the two also rebounded, also provided a protection so that more glucose could be taken up. This all means that there's greater insulin sensitivity if there's greater glucose uptake. And the final piece is that if you look at these bars, there was something peculiar. I mentioned this in the long form video. There's something peculiar about palmitate in that the basal levels of glucose uptake were increased. It seems like there was an effect, an increase. It could have been an artifact, something along those lines, or it's a real world effect. And I mentioned that it would have been cool. I wish that the researchers had done this, where they'd done statistics to look at the basal rates between these different conditions, because it certainly seems that the palmitate exposed cells in these L6 skeletal muscle cells had an increase in glucose uptake, even if it was blunted with insulin. And what do we find here? The exact same. Like, look at that. That is the exact same result in human cells, just as it is in the rat skeletal muscles. Now, again, granted, you have to double check this stuff for each one of the conditions that you look at. So if you're going to look at like Alzheimer's proteins, or if you're going to look at a cancer protein, look at MYC regulation, for example. You have to double check that all this stuff applies across the species. But for insulin signaling, it does seem that there's a great degree of crossover, even if the delta is different. Delta meaning the difference, the overall difference, or the amount of change. Okay, so in conclusion, I was debating even putting this in here because what's the point? But in some cases, it's context dependent. Animal cells are great proxies for human cells, or what happens mechanistically, specifically mechanistically within our cells. However, there are plenty of instances of differences, which I would totally check out that other paper that does discuss some of the differences. And there's a lot of other similarities as well that I didn't cover because I just wanted to focus on insulin signaling. So data should always be double-checked with human cells. This is definitely true if you're just looking at mechanisms, but certainly never making any major assumptions or major claims without looking at whole body measurements, which is what we've done as well when we've looked at insulin signaling. So I acknowledge that I made a mistake in that I wish, and going forward I will do this, that I will be presenting in human cells first, or human tissues, or human first, to establish a system, to establish a relationship between a treatment like saturated fat, stearate, palmitate, etc., and the actual effect. So do we actually see an effect with the addition or the exposure to this food? And then the question is, well, what is causing that effect? What are the mechanisms for that effect? And in that situation, I would still focus on human cells. But if we have an established model that shows a lot of similarity between animal cells and human cells, I will still be using animal cells because they give us great insight in a clearer sense, in a more manipulative sense for scientists, in a good way manipulative. I don't mean emotionally manipulative. I mean physically, like actually doing different treatments and changing them in certain ways. It's easier to do that with the animal cells. And if we know that there's a good that they are a good proxy between animal and human cells, then this will give us more data on the mechanisms, and I will flip how I do things. So humans first, then as we get into the mechanisms, I will introduce more animal studies. Okay, there we go. Hopefully you get some sense. I realize I sort of rambled. I probably could have cut this by like 15 minutes, but this is pretty much off the cuff. I didn't really have many notes with me. I just set this up and wanted to explain, so if in case anybody was ever wondering why researchers would use animal cells or animal models in general. Animal models are even more advantageous than animal cells in that they allow us to create what are called transgenic lines. So we can genetically manipulate the animals, and the benefit is that we can't do that with humans. So I'm definitely going to still be covering transgenic models for sure in animals. But again, still, you can never then assume, oh, well, this definitely applies to humans, because there are many instances where it doesn't, and yet there are also instances where it does. And I ran across an article that actually broke down all these instances where animal models were extremely helpful and did carry over to humans. So if you're interested in that, I'll post these links so that you can check it out for yourself. Anyway, a bit off the beaten path, but next week we will get back on the path, and I'll be presenting some really cool data for some follow-up studies, probably in human cells. I have this lengthy thing on mitochondria I'm thinking about doing as well. So I'll leave it there. Thanks for stopping by. Thanks for getting to this point if you did. Thanks for entertaining my rambles, and with that, I'll catch you in the next one. See ya.